For centuries, humans have been treating bacteria like germs to be eradicated. But what if we could treat them like our partners instead? That's the very question that Jared Kehe, Chief Technology Officer and co-founder of Concerto Biosciences, is set to answer. He, Sherry, and everyone else at Concerto Biosciences developed the kChip. I can hold it on the palm of my hand, but it can track tens of millions of bacterial interactions in a single day. As you'll see through this interview, Jared started out in skincare, but as he developed the kChip, he realized the potential it had to study all microbiology, whether it's environmental microbiology or other bacterial diseases. So if you're a microbiologist studying the microbiome, or if you're a biotech professional wanting to see more of automation and machine learning at work, or even AI at work, this interview is for you, and I hope you enjoy, because there'll be many more interviews like these to come. So first off, what about the human microbiome requires us to track so many bacterial interactions in the first place?
Think about a city of people. Think about New York City. Millions and millions of people walking about every single day. The city will produce some kind of measurable output, right? The total amount of work that the city is able to accomplish, the building of new infrastructure, so on and so forth. And we know that all of those outcomes stem from all of these individuals working together. So that's kind of one way to think about complex systems, when you look at them sort of holistically. And we do that with microbiomes all the time. We do that with the gut microbiome, we do that with the skin microbiome. We say, hey, here's 1,000 people with atopic dermatitis, here's 1,000 people without atopic dermatitis. Let's look holistically at their microbiomes and see if we can see the healthy microbiome somehow performing, protective functions for the host, and the atopic dermatitis microbiome is not. But we also know that those functional outputs, if you will, stem from these individual strains all working together in some capacity. Where a lot of microbiology traditionally has gone over the last, say, 100 years, is to try to pluck organisms out of the wild, whether it's a microbiome or a soil sample, what have you, and study it intensely. But if we think about our city analogy again, we know that human beings, when looked at individually, say you spend an evening by yourself on the couch watching TV or reading the book or taking a bath, the things that you do, your behaviors, your functional outputs are going to be very different if you're by yourself than if you're with a group of friends. With even one other person, suddenly you become a different person by virtue of that interaction. And again, the functional output of that pair is going to be very different than if you're by yourself. And so what Concerto is really interested in is, when do we begin to see, if we study groups, pairs, sets of 3, sets of 4, the emergence of those kinds of functions that we're observing when we study the system holistically? And can we translate that kind of insight, that kind of knowledge, into the development of microbiome altering products, direct-to-consumer products, therapeutics, and so forth?
I think you said in your press release you've got 33 million combinations in 5 months, and you've been able to screen at peak 1.4 million of these combinations in a single day. And to me, that's crazy, because that's like half as many users on the dating app Tinder, for one. And that scale is just like mind-boggling to me.
33 million is huge. And we're proud as an organization to have achieved that. The 33 million data points was the culmination of a almost decade-long dream to achieve screens of this scale. We began working on the kChip system in academia out of Paul Blaney's lab at MIT and the Broad Institute as early as 2015. At Concerto, we introduced a lot of additional innovation, predominantly on the automation side, to scale this system. To be totally clear, when we think about ecology, the number of pairs among those 1,000 is something like 500,000 possible pairs. Okay, that's a big number, but when we're talking about kChip scale, we could reasonably construct, using kChip, all 500,000 pretty readily. If you get to sets of 3 among your 100 or among your 1,000 microbes, we're talking about almost 200 million possible sets of 3. And if you get to sets of 4, we're talking about like 40 plus billion possible sets of 4. And so kChip is a huge step forward in our ability to profile combinations, but it doesn't quite approximate just how huge the combinatorial explosion really gets. So Through the lens of predictive modeling, understanding of complex systems like microbiomes, that's increasingly how we think about the utility of these big data sets. Yes, there's immediate applicability in looking at the 33 million data points and saying, well, here's a combination with these desirable properties. Sure, but think a little bit bigger. With 33 million, you can predict the behavior of millions of untested combinations that expands the space that you're looking at to discover novel biology quite a lot. So you have to put a lot of data in to predict that much more. And now we're talking about really understanding the full complexity of these systems.
Walk me through the hardware and the software that you needed to make this machine learning model work the way you wanted it to.
What exists at Concerto is all the hardware to actually generate the data in the first place. So in our lab, we have the kChip device, we have the microscopy setup, we have a lot of robots that move kChips around and load them with the microbes and move them over to the microscope and then move them into incubators. Then what happens is something that we call beaming, where all of the data generated on the microscope's computer beam up, abstractly, to the cloud. They download all of the raw images generated by imaging kChip. And at this point, it's literally micrographs, images taken on the microscope of the microbes in their little tiny microwells with some sort of optical readout of their behavior. So one of the most important things that kChip requires is a way of actually measuring function, a way of measuring behavior. So we might spike in, say, a fluorescently labeled pathogen that lights up as it grows, and we're looking for the microwells where the amount of emitted light is the least. That might imply that the microbes in that microwell are doing the greatest job suppressing that microbe. But to translate the raw image now into a numerical score that describes how well that function was performed is an image processing task. So what happens next is all of these images, and say there's maybe on the order of hundreds of microwells per image, and then there's hundreds to thousands of images, all of those images need to be processed. And so we have a dedicated individual that just figures out how to extract the numerical performance values from every single well. And then we have another individual still that will receive the spreadsheet of all of those different combinations. Here's a microwell from this image, from this kChip that had these microbes in it, and this is how well they perform that function. And this person's task now is to try to extract, commonalities, rules, patterns in that data. Their job is to rank the hits, which combinations perform this function best. And so it's definitely this kind of incredible process that goes from ideate the screen, execute the screen, collect the data, beam the data, analyze the images, summarize all the data, then do complex data analysis on that data to understand the ecology and rank hits. So lots of hardware, lots of software, lots of people involved, at every step of the way.
With the age of AI, do you anticipate this whole process could be automated, or are the people there also really important to just make sure everything goes smooth? It sounds like you've got a lot of steps, like that famous line, like beam me up or something like that.
That connection to Star Trek is not lost on us. In fact, it was probably purposeful. It's a great question. And as we conceive of larger and larger and larger screens to probe ecology that much more deeply, automation helps a tremendous amount. By virtue of automating kChip to date, we've unlocked something like a 5 to 10x improvement over what we were able to do when it was purely, run by human beings. Our next major step is to introduce additional kinds of ways of integrating and then replicating the totality of that automated setup. We're more or less at a stage where a lot of these individual steps or modules are automated, but what if you could press a button, and this of course oversimplifies it, and the total workflow from get the microbes out of the freezer all the way to beam the kChip images to our data team, what if all of that was just handled by one complete automated setup? To get there, let's be honest, that is a tremendous amount of human work. So a lot of the human beings that work at Concerto today are thinking not only about experimental execution, but how do we work really rigorously to prepare for the screens of tomorrow? And that's our great dream, is that the utility of this technology can extend its value, its utility through collaborative efforts with other organizations. One of the core inputs of a kChip screen is a biobank of strains. There are some thousands, tens of thousands from the oral microbiome, the vaginal microbiome, the soil, the ocean. Can we collaborate with organizations that have built these super impressive biobanks, run ultra high throughput combinatorial screens to help, us all understand how this incredibly complex microbial world works? Every time we churn this platform, we want it to be getting better, faster, in some way, shape, or form, incorporating new measurement capabilities. Another key aspect of what goes into a kChip screen is the function that you're measuring. kChip requires some kind of way of assaying or measuring the performance of each combination. So when we go out into the world and we talk with other organizations, the questions are always around what microbes are we putting into this screen and what's the function that we're going to measure for each of those combinations, and how do we measure that function? We've been fortunate to be able to work with corporations spanning food science to consumer skincare and everything in between to sort of dream up really fascinating ways we can study microbial ecology and develop products based on that understanding.
It sounds like you're asking not just how bacteria grow, or how quickly they grow, how much they grow, but you're also measuring the presence of different toxins, or molecules of interest that these microbes might be releasing. Or maybe if you're a synthetic biologist, you're trying to clean up an oil spill, you want to see how quickly though your microbes can metabolize the hydrocarbons and just break it all down. It reminds me of another story a while ago where I interviewed someone from New Zealand, and he said something about being able to discover bacteria that could degrade plastics. So you could theoretically also track how bacterial interactions affect plastic degradation just by measuring plastic concentrations over time.
Yep. So let's take that as an example. We want to first design a way of measuring that key function on kChip. So plastic degradation. So you could imagine perhaps putting in some sort of microplastic bead into every single one of our co-cultures, and we can directly measure for a screen that say is 1 million different combinations, how well do each of these microbes in combination degrade these little plastic microparticles. Then we can feed that data into the model, and the model can learn enough from that sort of preliminary foundational data set to interpolate, well, to do this most optimally, these are actually the microbes that you should use. So the understanding that the model generates about ecology through the totality of information that we constantly feed to it allows us to seed that model with any kind of new hypothetical function, run a few experiments, and then the model will do the rest to sort of look out across the total landscape of microbial ecology and point to the areas that we should really be looking.
You mentioned fitting AI models and basically developing them. Along the way, there could be the risk of the model being too simplistic or too complex, known as underfitting and overfitting. Correct me if I'm wrong. So did you encounter any issues either way? And which one did you see more often, and how did you address those issues?
So before we started developing these kinds of AI models, I think there was a lot of uncertainty. An open question in the field generally: can you ever predict how microbes behave in combination? And if you could, say you can take for granted that eventually you could with enough data, how much data do you need in order to make accurate predictions? And so the first thing, I don't think we were quite so concerned with the nature of any failure mode, like underfitting or overfitting, so much as we were concerned with, well, at what point do predictions even begin to look accurate? If you train a model with 100,000 data points, could it predict the performance of a microbe that you did not test directly, or a combination of microbes that was not tested directly? Or do you need 1 million, or do you need 10 million? And so the kinds of analyses that we were typically doing is, by virtue of having this arsenal of data, we would train models on progressively larger and larger data set sizes until we maximized the predictive accuracy. And it took literally millions of combinations until the accuracy began to kind of flatten out, and there were no longer any improvements in accuracy if you trained the model on an even larger set of data, provided that you train the model with a given function in mind and you're predicting that same function. So there were certain constraints involved in us getting to this point where it was making accurate predictions. We had to predict a function that kChip had already seen before. We had to predict the behavior of combinations of microbes, the constituents of which had been seen on kChip before, even if that specific combination had not. And so within these constraints, we began to see accurate predictions in the low millions of numbers of data points that kChip had collected.
So you're more focused on just getting it accurate in the first place. So that's why you all those tests. And then when you saw the performance plateau, you knew that this is like the least common denominator for being able to accurately predict how well a microbe would interact with like another partner and see how they would grow alongside each other. And now they have the AI doing it. A person accessing your database or working with you wouldn't need to do the testing themselves. They already got the prediction from you. All you have to do is just test it and confirm it, as opposed to doing a whole new test, which saves a lot of time.
Yeah, so like an organization with say 5,000 microbes, and they're interested in developing a new product to encourage the growth of some plant growth promoting organism, comes to us and says, run a simulation, run all possible combinations from these 5,000 through your model and help us nominate the top 50 combinations for additional testing. We can go from a space where there's billions of hypothetical combinations down to a handful of testable hits.
I want to close off by talking a little bit about the Skin Universe project. If someone in kChip were to access your data set today, like I think we discussed it, like we could see growth curves, we could see physical chemical properties, you can see all kinds of biochemical data and predict how bacteria interactions can affect the specific outcomes you're looking at. Now, do you plan to make that database publicly available, or is this something that you could use to just foster collaborations with you being like the hub for it?
We think more about the hub model. We've generated this data set. We understand how a lot of canonical microbes in the skin microbiome are affected by combinations of other microbes. That has a lot of opportunity for the development of everything from solutions to atopic dermatitis, dandruff, skin barrier defects, redness, irritation, and sensitivity, body odor. The data set represents a foundational resource for microbe-based innovation in skin dermatology and skincare. And so most of our interactions to date have been with organizations that are excited to leverage that data set with kChip to develop exciting new ingredients. But it's also a very cool new foundational resource to understand microbial ecology of the skin and perhaps even more generally. And to that end, we're excited to potentially work with academic labs to scrutinize this data, ask questions of the data, try to extract and discern common patterns and principles about microbial ecology, not necessarily in the context of the discovery of a new ingredient or a new product. What have we learned about this complex set of human symbionts that lives on us? Help us understand these 33 million data points. To that end, we invite enthusiastically academic PIs and students to reach out to kChip because we're equally interested in those kinds of collaborations.
And like you started out with skin, but as we discussed, like you could branch out to could create a whole new database for any kind of project theoretically, like beyond the skin microbiome here. Like we talked about the environment, we talked about other parts of the human body, even I imagine even animal, like like working with animals too. Even like, is there one particular part of like a facet of like biomedicine or environmental microbiology that you'd like to explore?
That's a great question, and we always have to be careful as an organization not to bite off more than we can chew. We have positioned ourselves and want to be a platform organization, which means anywhere and everywhere that microbes exist, we want to be working in. If I had to choose Oh, it's so tough, Paul, about women's health. It's a wildly underserved market and microbial in its very nature. So we're excited to do a lot of innovation there moving forward. Other areas that immediately come to mind that really excite me, microbes are anciently associated with food through fermentation, but when food spoils, it's because of spoilage causing microbes. There's a lot of excitement to be innovative about the role of microbes in everything from food preservation to the introduction of nutritious properties in food, functional foods, people sometimes call these. The kChip began many, many, many years ago through small-scale studies on soil microbes. And that's always been a special one for me. I would love to understand more about the microbial complexity of soil. It's definitely one of the most incredible, ecosystems that exists on planet Earth, and it's responsible in many ways for the health of the plants that we eat. It's responsible in many ways for the climate in which we live. If we could work on this ecosystem, that would be particularly special.
I love studying like the freshwater microbiome, the soil microbiome too. Like in fact, part of my project was like tracking like contamination sources. So I love that you mentioned soil because there are thousands, if not tens of thousands, like even maybe millions of strains that we haven't even characterized yet in soil. I love that you have so many options with the kChip and that you could like explore practically anything related to microbial behavior thanks to your new system. And I love it. So before we go, I also know based on our previous interview, you talked a little bit about ENS002 and how you've got that entering phase 1 trials. So what's next for that clinical trial?
So the ensemble number 2, ENS002, is a combination of 3 microbes that we discovered to suppress Staph aureus virulence as a means of treating atopic dermatitis. This just completed its first in-human phase 1 clinical trial. Stay tuned, Paul. We're going to be putting out a press release in the not too distant future to show the results of that trial. They're very exciting, and I'll leave it at that.
Once it comes out, I'd love to have another chat about that at some point.
Sounds great.
Awesome. Thank you for sharing everything about your kChip and the potential it has to transform how we do microbial research. Like the potential is just immense, and I'm really excited to see what comes up next for you.
Much appreciated, Paul. It's been fun to nerd out with you.